بسم الله الرحمن الرحيم. طلابنا السلام عليكم. إن شاء الله اليوم نكمل محاضراتنا بالاندوكراين ومحاضرة الاندوكراين بنكرياس، وهي المحاضرة السابعة في الاندوكراين. The pancreas is composed of two major types of tissue, the including of acini, which secrete digestive juice into the duodenum, and the islet of the pancreas, endocrine pancreas. The endocrine cell of the pancreas are arranged in clustering, and this is called islet of Langerhans, which compose of 1% to 2% of the pancreatic mass. There are approximately 1 to 2 million of islet of the pancreas of Langerhans inside the pancreas, each about 1.3 millimeter in diameter. These islet of pancreas, of Langerhans, containing of 4 types of cells, including of each cell secreting of different hormone or peptide. The beta cells compose of 60% of these islet of Langerhans and secrete insulin. The alpha cell composed of about 25% and secrete glucagon. The delta type represent of about 10% and secrete somatostatin, which is the PP cell. This present in a small number in the islet of the pancreas that secrete hormone of uncertain function that's called pancreatic polypeptide. So it is called PP cells or polypeptide cell. And these the presentation of the pancreatic islet of Langerhans. We see this is the exocrine function here. Good. Now, what is the structure and synthesis of the insulin? Insulin are prepared as a pro hormone, then cleaved in the endoplasmic reticulum to form the proinsulin. Most of this is further cleaved in the Golgi apparatus to form insulin and C-peptide fragment before being packaged in the secretory granules. إذن يعني حتى يتكون أول مرة عندنا برو هرمون الاندوبلازميك ريتيكولم يتحول إلى برو إنسولين، والبرو إنسولين بعدين يتحول إلى ينقسم إلى إنسولين والسي بيبتايد، يعني يطلع إنه إنسولين ويا سي بيبتايد. فأنت حتى تريد تقيس ممكن تقيس الإنسولين ممكن تقيس السي بيبتايد. Both insulin and C-peptide are secreted together. C-peptide may serve as a protective rule, helping to prevent renal, neural, and microvascular pathologies. Urinary excretion of C-peptide is useful marker of insulin production. لما قلت أنا بساعة لما أريد أعرف يطلع عندي إنسولين لو لا، أقدر أعرف من خلال قياس السي بيبتايد اللي يطلع إنه وين باليورين، على اعتبار إنه البرو إنسولين من ينقسم أو يتحول راح ينطيني إنسولين وبنفس الوقت يطلع وياه السي بيبتايد. هاي الكمية تساوي لنا هاي الكمية. هذا الشريط اللي يمثل لنا structure of insulin. The connected C peptide is cleaved to form the insulin. Specifically, insulin excreted is important action on the following tissue. So the main function and the action of the insulin, including of this tissue. First, liver. In the liver, increase the glucose uptake and also increase the glycogenesis, formation of glycogen storage from the glucose. Increase lipogenesis, formation of dry glyceride, the storage from the lipid. The action of insulin on the adipose tissue. First, increase glucose uptake, increase free fatty acid uptake, and increase lipogenesis. The effect of insulin on the muscle. Increase glucose uptake, increase in the glycogenesis, increase in amino acid uptake, and also protein synthesis. Insulin is the only hormone that lowers blood sugar. Epinephrine, growth hormone, cortisol, glucagon increase blood sugar. It does so by stimulating the uptake of glucose from the blood into the liver, adipose tissue, and muscle. This glucose is first used as an energy source and then stored in the form of glycogen in the liver and in the muscle. Excess glucose is stored as a fatty acid in the adipose tissue. Insulin is a major anabolic hormone which is secreted in response to the carbohydrate and or protein containing meals. Anabolic hormone tend to promote protein synthesis, increase the lean body mass. Other anabolic hormone including thyroid hormone, growth hormone, or insulin-like growth factor, and sex hormone. What is the metabolic action of insulin? The metabolic action of insulin, effect of insulin on the carbohydrate metabolism. Ethernal carbohydrate. Insulin increases the uptake of glucose in the muscle and fatty tissue by directing the insertion of glucose transport, gluco4, into the cell membrane. After that, insulin promotes the formation of glycogen from glucose in the liver and in the muscle by increasing the activity of the enzyme glycogen synthase. Insulin inhibits the glycogen or the glycogenolysis, glycogen breakdown. Insulin also promotes conversion of excess glucose into fatty acid and inhibit glyconeogenesis in the liver. What is the effect of insulin on fat metabolism? Insulin increases the storage in the adipose tissue by the following. Insulin increases the utilization of glucose by most of the body tissue, which automatically decreases the utilization of the fat, thus functioning as a fat spare or sparer. Insulin promotes the conversion of glucose to triglyceride. Insulin decreases the triglyceride breakdown and lipolysis in the adipose tissue by decreasing the activity of hormone-sensitive lipase. This enzyme is activated by stress hormone like cortisol, growth hormone, epinephrine, and glucagon. What is the effect of insulin on protein metabolism? Insulin increases the protein storage of the body as the following. Insulin stimulates transport of amino acid into the cells and also increase the translation of the messenger RNA and also increase the rate of transcription of selected DNA and inhibit the catabolism of protein. Insulin effect on potassium. Insulin promotes potassium movement into the cells, or probably by increasing the activity of sodium-potassium ATPase in most body tissue. This potassium-lowering action of insulin is used to treat acute life-threatening hyperkalemia by stimulating or simultaneous administration of insulin and glucose. The important things we have to know is how the insulin functions or how the insulin is secreted, so the mechanism of insulin secretion. Transport of glucose to the beta cell is usually by glyco-2. الحقيقة من يجي الإنسان يبدي يأكل ويرتفع عنده مستوى السكر، هذا السكر راح ينتقل من خلال الناقل كليكو ترانسبورت 2، ينقله وين يوصله إلى داخل البنكرياس، ويوصله إلى داخل البيتا سيل. Once inside the cell, glucose is phosphorylated to glucose 6-phosphate by glucokinase, and the glucose 6-phosphate is subsequently oxidized ATP, one of the product of the oxidation process or oxidation step. ATP closes ATP sensitase or sensitive potassium channel and depolarize beta cell membrane. Depolarization of beta cell membrane open the voltage sensitive calcium channel, and calcium flow into the beta cell down its electrochemical gradient, and the intracellular calcium concentration increase which cause insulin secretion by exocytosis. إذن العملية ببساطة إنه من يصعد مستوى السكر راح ينقل هذا السكر للبيتا سيل، البيتا سيل راح تصير عملية الفوسفوريليشن باي ذا جليكوكاينيز، راح يصير عندنا اوبننج إلى channels يدخل الكالسيوم والكالسيوم concentration increase، وراح يفتح لنا عملية خروج الانسولين. لحد هاي اللحظة إن شاء الله خلصت المحاضرة مالتنا. نكمل بقية البنكرياس اندوكراين إن شاء الله في المحاضرات القادمة إن شاء الله.